Hello friends, we are going to learning the mechanism of enzyme action. The mechanism of enzyme action was firstly studied by the scientist Michaelis and maintained in 1913 during the hydrolysis of sugar by invertase enzyme. Basically, we knowing that the enzymes are the proteins, complex proteins, and they are having the active sites. So with this active site, the substrate get bind to the enzyme and enzyme substrate complex get formed, and finally the product is liberated. The enzyme escape from the reaction. So enzyme substrate reaction is completed in the two steps, that is step first, enzyme linked with substrate to form the ES complex, enzyme substrate complex, and the step second, from this enzyme substrate complex the product is get formed and enzyme get liberated from the reaction. So the rate of reaction is controlled by different factors such as the enzyme concentrations and substrate concentrations. So these two factors are called as limiting factors, and it is very affecting on the reaction. So rate of reaction is depends upon the many factors like temperature, pH, allosteric modification, feedback inhibition, then successful collision between substrate and enzymes. Also, it is depends upon the concentration of substrate. So see here, this is the graph which showing the Vmax and Km values of enzyme. So we knowing that the velocity of reaction is depends upon the substrate concentration, that is S, substrate concentration, that is the S. At a low concentration of substrate, the initial velocity or rate of reaction gradually increases, but the concentration increases beyond some limit, the reaction gets saturated, reaction gets slows down, and the velocity of reaction remain constant. This point at which the velocity of reaction remain constant is called the maximum velocity or Vmax. So see here, the all enzyme molecules are utilized here for formation of enzyme substrate complex, due to which there is no further increasing in the rate of reaction takes place. At this stage, the equation for velocity curve is as V0, that is initial velocity, is equal to Vmax into substrate concentration divided by Km plus substrate concentration. So what is the Km? So Km is nothing but the Michaelis constant. You can look here the graph. So graph showing that the V max divided by 2, that value is nothing but the Km value. So the substrate concentration at which the half of reaction, that is V max by 2, is completed is called as the Km. So the Km is also called Michaelis constant. So substrate concentrations at which the half of the reaction get completed, one can be say as The substrate concentration at which the half of enzyme molecules forming ES complex molecule. So kinetic behavior of enzyme is identified with the help of Vmax and Km value. So smaller Km value is indicating greater affinity of enzymes to the substrates or vice versa. So what are the significance of the Michaelis constant? So Km value is inversely proportional to the enzyme activity. The smaller Km value showing higher enzyme activity or higher Km value showing smaller enzyme activity. The smaller Km is greater affinity of enzyme to the substrate and vice versa. Km value shows variability and affinity to the enzymes. So Km value is used for identifying isoenzymes. The Km is used for determine the actual active sites present in the enzymes. So this is all regarding the mechanism of enzyme action. Now we will learn there are two mechanisms of enzyme actions, that are the log K hypothesis and induced feed theory in incoming lecture. Thank you very much.